Our second speaker today is Derek Toomre, who's professor of cell biology and director of the Yale Cinema Microscopy Lab. He received his PhD from University of California as an expert on quantitative live cell imaging, and he specializes in innovative approaches to microscopy, including fluorescent probes, data analysis, and spatial mapping. He received an NIH Director's New Innovator Award and a Calvi Fellow, and today he's going to talk about his recent work, which is primarily focused on spatial control of membrane trafficking during cell morphogenesis, migration, and cilia formation. So Derek, the floor is yours. So Derek, you're muted.
Okay now? Yes?
Yeah.
Okay, great. Let me go back a slide or two. Great. So I'll be talking about cilia, which have some relevance to cancer and signaling. I'll be talking basically about a new pathway that we are discovering and tell you about the ramifications of this. I'm not talking about any work on SARS-CoV-2, though I do have some work on that, and my conflict of interest is declared there. So in terms of sort of a primer, let me just sort of tell you the main message. And the main message is that we're proposing and characterizing a novel pathway of cilia delivery to the surface. And I will sort of tell you what basically the central paradigm is, and then go on to sort of describe to you that I think that that basically our data suggests that we have an alternate pathway here. Oops. Okay. Okay, it's clicking differently. So let me first start off with a quick primer here about primary cilia. So, really sort of two aspects here. What are they? And I suppose sort of related here, why do you care? Why should we care? So in terms of the first part, that's relatively easy. They're long, slender cellular antenna. They're basically present on nearly all cells. There's the exceptions being red blood cells and T cells. If you go by electron microscopy sort of light microscopy, you see they're long and slender. The width is maybe 150, 200 nanometers, and they can extend to about 10 microns long. They're solitary organelles, they're primary cilia, and let me tell you a little bit about some of the known functions. So you can sort of think of them as a specialized sensory antenna. So you have an adaptation of this for vision of your rod cells, I guess relevant to right now and coronavirus. They also important for smell, and there is a loss of cilia, which is causing the ciliary dysfunction of that, and as well as signaling aspects. And let me sort of mention a little bit more on that because it's really relevant to this audience, especially in regards to cancer. So primary cilia. So why do you care in this? And the easiest way of sort of thinking of this is thinking of in terms of ciliopathies. Okay, there's a number of diseases that are basically dependent. It was actually sort of discovered retroactively. These diseases have been known for quite a long time. And then only in the last couple of decades was it really realized, in part from pioneering work at Yale, about the sort of link between these diseases and disruptive ciliary function. So you can see this in sort of the Joubert syndrome or liver cysts, the kidneys. It's quite multi-organal. But again, for this audience, I think it was particularly relevant is in the context of cancer. And you can sort of think of it in terms of sort of two aspects. One, aberrant signaling, and then your cilia being part of the tool that would turn things on and off. It gets a little more complex here because you actually can release vesicles from a cell, which can transmit signals. So that's one aspect of it. But you can sort of think of it in a simplified way of controlling the signaling on or off. Now, just highlighting that complexity was sort of a review that sort of indicates that it actually can go either way. you could have a certain degree of activation. You could turn on hedgehog by activation of this receptor level in sort of pathway B, or you could actually do it by going independent of that. So if you look at cancer, some are activated by the presence of cilia and some are by the repression. Okay, but if we sort of go beyond that aspect and say, well, what is sort of the normal paradigm? Well, the normal paradigm here is that cilia occur once per cell cycle. Okay, so you generate them during G1, G0. They're sort of present throughout, and then they have to be assembled during division. So in that sense, if you sort of think of it from that lens, you're really not controlling it except for to the point of cell division. And maybe any delays on whether you would have ciliogenesis or not. And I'd like to, so say, change that paradigm view. So, reminding you again, I do a lot of imaging, as you sort of heard in the intro. This is just one example of super resolution imaging where we can actually see this very long structure of a cilia, which is, as I said, it can be 10 microns or so long, with a resolution down to sort of 20 nanometers, where you can actually see it as a tube. So that's the one sort of technique, but I'm going to show you other sorts of imaging techniques and sort of exemplify how that can provide new insight here. Again, this is sort of the structural aspects of this axoneme. Sort of an aside, which is relatively relevant, is that they can present with this pocket. Let me see if I put the laser pointer on, and in the ciliary pocket here. And this is sort of shown extra short here, but this can be a very deep invagination. In fact, it can almost be nearly entirely invaginated. Okay, so what is sort of the key process of how they form? So the general paradigm, in fact, there's one for epithelial cells, which I will not sort of discuss. But for most other cells, in fact, the majority, they start with a ciliary vesicle, which forms, and then you have this double membrane, which sort of bends in. And then you actually have this really, think of it as a super large vesicle. It's a vesicle, but this vesicle could be 7 microns long, okay? And then as an event, it actually has to go and fuse with the plasma membrane, produce. This is a standard paradigm. This happens once per cell cycle would be the standard paradigm. Now, I want to bring in one other player here, and this is one that we've worked with quite a bit, which is called the Exocyst complex. It's a tethering complex. It was first discovered actually at Yale in yeast, and basically it is known to basically drive the upstream of sort of snare fusion machinery to allow spatial temporal control of vesicle exocytosis. Now, this, as you can sort of see in this review, has been thought to, for quite some while, play a role in cilia, namely ciliogenesis and stabilization, by targeting the vesicles right here where they would fuse and then basically drive, let's say, control within the axoneme. That is the standard paradigm. And while I don't want to say that is wrong, I think it's actually missing, let's say, 80% of the picture. Okay, so let me sort of go back to why we have this paradigm and what's sort of the underpinning it. Well, is it I want to basically first say that it's based on indirect evidence. We've actually not seen the vesicles there fusing there, nor actually seen much of the exocyst. And I would say that you're missing something that's really important and is incomplete. But let's sort of get beyond that and let me show you what I'm going to be talking about. But I first have to tell you why, if we're claiming to see something different, why and how are we able to do so when people have been looking at cilia for quite a while? And so there are a number of sort of technical aspects to the solution. The first one is we're using a technique called total internal reflection fluorescence microscopy. It's a axial super resolution technique, and it allows us to image just the lower surface of the cell and actually see cilia's emergence. Okay, but that actually, even with that super resolution technique, is not enough to be unequivocal there. the distance between cilia and the surface at times is very low, and it's actually not clear if they're inside the cell or actually have emerged and are outside. And think of it again like your antenna. Your antenna, if you sort of just give an analogy, is going to respond to signals quite differently if it's actually outside your car, truck, whatever it be, versus pulled inside where it cannot receive the signals. Okay, at least not the same signals. The other technical aspect is we used we'll call it clever pH switching to identify when cilia are in or out. We're using this as in a pulsed way, and we do molecular replacement of the exocyst. The latter is important because for a long time people were imaging the exocyst, and they were just simply overexpressing it, and they would sort of see localizations like this everywhere or some accumulations. But when we did this replacement strategy, we could see it in these discrete punctae. Okay, and this is going into HeLa and other types of cells, looking at vesicle exocytosis, and we could see in these chymographs distinct events. And I just show you one trace where a vesicle has arrived, as we see with the exocyst, and then with another reporter, and this is a snare reporter here, which is a fluorine with the pH sensor. We can actually unequivocally identify the fusion event. So this is sort of a constitutive pathway. We know it's coming from recycling vesicles, and we can identify and study that. Okay, and we can tell the events about when the exocyst appears and when you have the fusion event. This is relevant to how we're going to be looking at things with the cilia. So what is it that we see in, just a second. Okay, good. Okay, so here's a short movie of what we're seeing in terms of cilia, sort of called the biogenesis aspects, where we see basically exocysts recruited to cilia. So that's sort of standard. That itself is basically showing it going to the base of elongating cilia. But there's actually another phenomenon that we observed, which is quite different. And so now you actually see the cilia with, the reporter here is smoothened fluorine, or we can also use smoothened GFP. So that looks a sort of characteristic curvy linear cilia. This would be in the dimensions several microns long. And what I'd like you to note, and I'm going to play this more than once, is that we actually see the red signal getting recruited to this cilia. Boom, right there, off, on again, off again, several times. And this is the time, one last time, is in hours, okay? So there's no signal there, it appears, it disappears, it appears again, over the course of, in this case, of this movie, which is basically 4 hours. So, and appearing there in this sort of the minutes range. This does not fit with what you would expect of sort of a couple of things. One is it's along the entire cilia, not just the base. Two is it appears there, goes there, and then vanishes, and then comes back, an hour or two later again. So what's going on there? And what do we, how can we sort of probe into that? With, I guess you can intend the pun. So we do this by using, again, we're using this Exocyst, and we look after stimulation. We see something interesting happening here. And you actually see it quite early in the movie. Let me just play it again. And you see, bam, I realize it's hard to catch. Bam, you see that green object? That's the vesicle flying off, okay? So we see the release of the vesicle happening as well. Now that's actually, that part is known, but later we actually see then the exocyst appear, okay, after we've dropped off that signal. So basically the SecBRFP, which is an exocyst reporter, decorates cilium after serum stimulation and vesicle release, okay? And by the way, we artificially, to generate cilia in tissue cell culture, starve them. Of course, the normal situation would be in serum, okay? Okay, so, per that sort of model, well, is there any evidence for this? Yes, there have been some papers here. And it's, you call it, they call it, the word is, they call it decapitation, okay, where it cleaves off that vesicle. We actually think that the mechanism is going to happen is different, okay? They are releasing it, but you'll see in our cartoon at the end, we're thinking that it's happening by a different mechanism. You can obviously see sort of the importance of that on signal transduction, as I was basically indicating earlier. So what do we happens to our Exocyst reporters after we add serum, in this case, FBS? And so what we can see here, and here's sort of the overview slide, we are using Exo70 as our Exocyst reporter, IMPP5E is a Cilia reporter, and a couple of different things. One is, and I should mention, first of all, this is endogenous Exocyst, it's no longer replaced. Okay, so this is sort of native conditions. And what you can see is you can very clearly see it localizing to cilia. But if you look carefully, and I think you can see in this case here, and in this case here, it actually, it localizes there, but also forms these additional tubes, which are pulling, well, look to P, and for movies, we would know, are pulling out of it. So what's going on there? And what I will propose to you and submit to you is that this cilia is actually inside the cell and is actually being remodeled by the exocyst to pull off other membranes and remodel it to actually have the role of exocyst being to tether and help fuse this monster huge 5-10 micron long vesicle. We basically see this, and here's sort of another view of that. And again, there would be fluorine smoothened there as well. So you smoothen IMPP5E and these large tubules that are pulling off. Now, does this happen when you stimulate with serum? And the answer is yes. And if you look at the percent of cilia, you add serum, they drop down a bit. That's expected. What is particularly interesting and exciting to us is that the colocalization with Exo70, which is usually quite low, goes up dramatically by adding serum. Okay, so serum is going to turn over this also with the cell cycle, but the important thing is that's actually driving that recruitment. Now, we did some controls here. If we knocked out the other Exocyst complex members, okay, Sec10 or Exo70, we do not see that. Another way of saying this is if we destabilize the Exocyst, so you don't have your entire 8 protein complex, then we no longer see that localization. Okay, so that's sort of a control experiment. Then you might ask, well, okay, so it's recruited, but where is it being recruited relative to the Exocyst? Relative to the cilia. And I kind of reminded you early on that you have this ciliary sheath. It's sort of a membrane which kind of does this. Let me put the pointer on. You have the membrane here, and then it's a double membrane, right? So it kind of goes like that and curves all the way around. Okay, so or you can kind of see it here in this cartoon. And is it here, the Exocyst there, or there? Now, there's sort of different reports. There's obviously the ones of it on the base. There have been some reports of about being inside, but we actually believe it is actually here along the pocket. And how do we know that? By using a super resolution imaging modality called structural illumination microscopy. And I think you can sort of see it easily here. You'd have smoothened, so this would be basically your cilia reporter, which would be on the extracellular side, or I should, right, so it'd be, on the inner membrane, if you like to look at it that way. And we see Sec8 or Exo70 as a wider distribution. Okay, so it is on, if we're going, let me turn off my pen here. If we're going back, it would be on the ciliary pocket membrane. Okay. We switch to pen. So we see these tubules emerging. I showed you one example, and we actually see it in live cell imaging as well, where these tubules are dynamically pulling out of cilia. Okay, so we think it's actually important for the remodeling. Okay. It's a little funny in giving this presentation, switching back between the pens and not, but let me just tell you to sort of focus on here on the point of the arrowhead, where we actually think of that as a fusion event. So you have this internal cilia, the cilia which is inside the cell, and is here, and you have SecBRFP there on it. We think it's important for then the tethering function. And at this point right here, where the membrane, it looks like it expands, you see that little bit of a burst. At that point, the SecBRFP collapses to the base, okay, which has been sort of purported in the literature to exist most of the time. Again, at a single snapshot, you'd mainly see it at the base. But actually, if you look at it live over, let's call it longitudinal, you would actually see it earlier on these internal cilia. So we actually think it's driving that process out. Okay. Well, how can we actually sort of prove this unequivocally? And the way that we've done this is basically using a pH switching experiment. And when the cilia are outside, if we switch the pH and we have this pH sensitive reporter, every time we switch it, it goes on and off. Okay, so we make it acidify, we can turn it off, and then it goes back on again. So we basically, by switching the pH, we can tell if it's outside. Here's switching going up and down. But as you can see in this context, it's not. It's actually resistant. And why is it resistant? Because it's inside the cell. So it's basically inside, inside, inside. And then here you can see it switch into the purple or magenta, indicating it's now outside. Okay, so there we can really say unequivocally that it is actually merging over time. And you can sort of see these experiments. We're switching the pH, the intensity goes up and down, the SecBRFP doesn't change. Here is a case where it's actually not changing much. The thing fuses, right, sort of, it's not switching much, it fuses, now it starts switching, and then just a little bit later, you see the Exocyst, it's actually changing to part of it, and then it eventually drops off and fully into the base. Okay, so we can start to identify what machinery is there, sort of looking at who might be the players that might be engaging with the Exocyst, and there's some interesting ones such as RAB10 is a likely player, and actually we see RAB10 localizing there, which is actually different from some of the other RABs. So I'd like to sort of end now in the remaining minutes with basically the working model and just walk you through that very briefly. So the working model is that in the starved state, you have cilia with these sort of deeply emerged pockets. The Exocyst is in the vicinity of these recycling endosomes, and probably you have some targeting of vesicles here. In the serum stimulation event, which is actually the normal case, right? If you sort of thought about cilia in the body, they're actually constantly being remodeled. Okay, so the event of releasing the vesicle is actually different than the sort of what was proposed, we believe, of it sort of coming off from one that's fully outside. Is actually, as it starts to pull in, we believe that there's remodeling that can actually promote this vesicle to release. Once inside, you have recruitment of the Exocyst to this large inside vesicle, which is again the majority of this sort of internalized cilia, which can remodel pulling off tubules, and consequently then can recycle back. So you have this entire pathway here that can modulate the signaling. So with sort of that, this is, oops, switch back to automatic. And really this is driven by senior scientists in the lab, Felix Riviera Molina, supportive of other people, but really he took the lead here. So that's where I'd like to end and address any comments that you might have.
Thank you for a very interesting talk. Are there questions from the audience?
Let me pull up the chat.
And you can just type them in. While we're waiting,
Yeah.
so there are a lot of genetic disorders of cilia formation that have many different phenotypes. Are any of them associated with increased cancer risk?
Yes, there have been, and we're sort of where it gets complicated is it depends on sort of what cell types you're looking at. Again, pushing things up and down, as sort of mentioning earlier. So yes, there are. Haven't been sort of investigating this, so much personally, but yes.
Okay. Thank you. Questions for Derek?
I know this Zoom atmosphere makes it a little bit different. I'll enjoy when we see people face to face again.
Another question then.
Mm-hmm. Sure, Dan.
In cancer cells, which are obviously many of them often constantly proliferating, do you see abnormalities of cilia formation? Do they have more cilia? Do they turn over more rapidly? What happens? Is that known?
Right. So this was a little bit what I was alluding to. You can kind of push it either way. So that's actually where the confusion is to cancer, is that you would say, well, do they have more cilia? Do they have less cilia? And basically the paper review had indicated that there's sort of the two sides on it. So in one case, you actually hyperactivate by, let's say, adding smoothing and hedgehog signaling. So that hedgehog is obviously a key component. Same with sort of PGGF. So it'd be basically a hyperactivated. The other case is where you would actually activate the signaling by the absence of the cilia. So that's sort of where it's, in some cancers are driven by having cilia, and some are driven by the absence. That's sort of given the funny paradox, depending on that nature of which signaling pathway you're talking about. Is it a hedgehog sort of smooth-in type of one, or other signaling pathway? And what is the activation at the basal state? So that's why I think it's given some complexity to the field, because you couldn't just simply say, this is only this way. But it does, there's evidence for both there.
Perfect. Are there any other questions from the audience?
It's getting
okay. If not, thank you all for coming. Thank you for two speakers. It was very interesting. And everybody, you have two extra minutes for your day. Thank you.
Thank you, Dan.